to use the key. Let me just quickly test. Okay, this is some of the latest work that we've been doing recently, and so this is really it's really fresh off the blog, so it should appear anytime soon in infectious diseases. So, but yeah, I mean, it gives us direction for unexpected discoveries. So, we don't even. So we are seeing an unprecedented rise in superbugs. So that's a term that we are consistently hearing, and these superbugs mainly are connected with hospital-acquired infections. So what we are seeing is that if immune-compromised patients or other patients acquire these superbugs, the death rate is as high as 50%. And what is more alarming is that last year, about 16 deaths were reported over a 4-year span, just by being infected by one particular strain of a bug, so which is Klebsiella. So they die of pneumonia, but it's literally alarming. The reason why they are called superbugs is because of their ability to be resistant to antibiotics, not just one antibiotic, but to multiple antibiotics. And if you see that this problem is not just restricted to one place. This problem. I mean, this cutting is from Australia. So you go anywhere and you find this spurious bugs that are coming up that are really difficult to deal with. And one of the things that faces or that is a grand challenge for all drug discovery processes is basically to come up with a solution of how to overcome this resistance. So I just want to give you a brief overview of what these antibiotics look like. The ones that we are going to talk about, that I'm going to talk about today, is something that is called beta-lactam antibiotics. So this is the most widely used group of antibiotics. Penicillin, cephalosporin, carbapenems, they are all from the same class. The way they derive their name is based on this beta-lactam ring that is present, and they are basically the most commonly used antibiotics in infection. So the way they work is that they bind to this penicillin binding protein and inhibit peptidoglycan synthesis. So they are responsible, these proteins are responsible for the assembly, maintenance, and regulation of the cell wall, or the peptidoglycan. And once you disrupt that, the bug is not able to proliferate. So how does the bug acquire resistance to these antibiotics? So there are several ways. So for example, in one way where the antibiotic just doesn't reach the target protein and is just evicted using a porin, a cell-based, cell membrane-based protein, and is just completely evicted out. You have the inhibition of binding. So the protein that binds to these antibiotics, there's a modification in the active site. But the most common of the whole lot is using this enzyme called beta-lactamase. What it does is that it hydrolyzes that beta-lactam ring and completely inactivates the antibiotic. So the majority of resistance that you would see or you would hear about would come from this beta-lactamase, this enzyme. And there are several types of beta-lactamases which have different specificities for various different antibiotics. So here is just the common beta-lactamases that that are prevalent out there. And I don't expect you to read any of this, but just to give you a concise view is that when they were first identified, there was a broad spectrum, and then we saw that these enzymes began to hydrolyze a different and a larger variety of antibiotics. So expanded spectrum came through. Then you have this ampicillin type of molecules that are hydrolyzed, and then you have the latest generation which hydrolyze carbapenems. So which is the carbapenemase. So the ones that are we're seeing predominantly as superbugs. So how are they actually transferred? So the gene for these beta-lactamases is on extra chromosomal DNA, so it's on a plasmid. So the plasmid carries the antibiotic resistance genes, and they are usually encoded on transposons, which are also mobile. So they can be transfected or can move around from one bug to another. And what we have seen is that they're just not restricted within the species, but you can see that TEM1, which is an enterobacteria, can also infect influenza and other different bugs as well. So the carbapenems, the one that we are interested in, are the latest generation of antibiotics, and that is why it is more alarming. Is that the latest drugs that are being licensed by the being approved by the FDA are also becoming resistant. So carbapenems, as of now, they are less affected by the common mechanisms of antibiotic resistance, but they are still. We've seen that in vitro there are different bugs that are completely resistant to carbapenems. So how do we get over this sort of mechanism? So what we are looking at are these enzymes, the machinery that catalyzes this reaction. So what it does is that this ring is split open and it's completely lost in this case. So there are two major groups of mechanisms. So one involves a serine that is in the active site, which is the A in class B, or you have the zinc. And again, they hydrolyze all sorts of antibiotics, including the latest generation ones. The one that we are going to focus on today is the Klebsiella pneumoniae carbapenemase, which is a class A. It is inhibited by clavulanic acid, but not by EDTA, which suggests that, of course, there is no zinc here, so the EDTA cannot chelate, or the other ions as well. And it confers resistance to all beta-lactam antibiotics. So what it does is that if you pick up a mutant carbapenemase, there's absolutely no antibiotic that is present that it cannot hydrolyze. So again, it's plasmid encoded, and it's resistant. It's associated with other resistant genes as well, and it's highly transferable. So this is, luckily there is a crystal structure. So there's a crystal structure available for KBC2. And so this is what it looks like. So it's roughly divided into two domains. So one is the bottom half, which is the alpha helical domain. And the top bit is more beta sheet that is linked by two alpha helices over here. So if you look at, if you have a close-up of the active site, which is in the lower half of this, you see that the active site is surrounded by 3 loops, and these are very, very critical for what we're going to go. And so the 3 loops are basically, which has this critical a tryptophan, which is 105. Then there's another loop called the omega loop, and there's a loop that is, again, the third loop that is positioned here, which we call as the hinge region, which is somewhere here, and as a result of that. Now, this green colored molecule is the drug. So there's a crystal structure with a drug that is present here. But if you go through the literature, you will see that there are a lot of Me Too studies there. So the catalytic chemistry is very well established. So whenever you see a new paper coming out, it's not very different from what is already existing there. So that's why one of the reasons why why we wanted to get away from this was to have a completely different radical approach to our program, was not to go ahead and design any of these me-too molecules as well. So what we decided to do was to go ahead and look at the inner regulatory mechanism, look at conformation and allostery, or how the active site is remodeled before we go ahead and do any ligand design at all. If you look at the literature, one very good example comes from this drug called PSR3226. What it does is that when it reaches the binding site, it covalently links to the catalytic residue, which is serine 70, and forms a trans-enamine intermediate, which is irreversibly trapped into the binding site. And what it does is that, as a result, it also stabilizes the local architecture, besides disrupting other interactions that are present there. And this is a very effective form of drug. But again, like I said, there are mechanisms through which the bug can override this mechanism. So we have to get away from this completely. So what we did was we pulled out all the structures that are present. So there are 3 structures that are present in the PDB, and what we did was we did the sequence alignment. Of course, we can't read this, but what I want to show is that if you go through the databases, you find that there are 83 non-redundant sequences that you can pull out that have a sequence identity of greater than 26%, and you see the colors that there's an extremely high conservation of residues in the structures. So again, if you look at the phylogeny, you see that most of them. So here's our carbapenemases. These are the other 2 structures that are present in the PDB, and you say that they're not much different, but they can completely align themselves. So when we do a structural alignment of CHEM1, SHV, and KPC2, we find that there are certain patches of conserved hydrophobic residues, 3, 4, 5, that are present either before or after these critical residues that are involved in catalysis, or in other structural positioning of the ligand, and you can see that they form a sort of a hydrophobic network. So we will call these green things as alpha net, whereas the top bits are beta nets. So you have a conservation of sort of a hydrophobic network that exists in the over the entire protein. And if you look at these bits, you see that they are not so conserved, but the hydrophobic nature of these residues are conserved over the entire 83 sequences or 83 bugs that we looked at. So then we asked ourselves this question: is that what are these hydrophobic networks? What role do these networks play? So we carried out a classical MD. So we wanted to look at the inherent flexibility of the. So we did MD simulations of the wild type as well as the one with the ligand. And if you look at the RMSF, you see that these are the 3 regions that are important. We see that in region 1, which shows very high mobility, is where the critical tryptophan residue is present. Whereas if you look at region 3, is this region, which is the hinge region, and region 2 is where you have the omega loop. But if you look at the drug, what the drug does is that you have a very different set of motions. You have a very high mobility in region 1 and in region 3, but nothing in region 2, because this region 2 is being stabilized by the drug that is present. So we then went on to do and the other thing that we saw from these from the PCA analysis was that we identified 2 different conformations of the tryptophan. The tryptophan is very important because this is what stabilizes the beta-lactam ring. So it does bi-stacking interaction with the beta-lactam ring and presents the carbonyl oxygen to S70. And we see that in the active side, in the crystal structure, you have this flipped out conformation, whereas when we look at the PCA, we saw that it could adopt another conformation, the flipped in conformation, which completely opens another whole set of a sort of an allosteric binding pocket that allows us to see that that we can look at interactions if we want to go ahead with design of new molecules. But then at the same time, what we were seeing is that in the crystal structure, there were these hydrophobic interactions that were being disrupted when the drug was present. So we asked ourselves this question: is that what is so important about these hydrophobic residues? So what we did was we then we asked ourselves a question that if we inhibit this, or if we design the loss of function mutation, so if we mutate these hydrophobic residues into hydrophilic ones, then what is the sort of effect we're going to see? So we designed, in effect, we were trying to disrupt this hydrophobic network to see how the protein was functioning. So we then carried out enhanced sampling in order to understand what is happening. So these are the collective variables that we defined based on the three important residues that we were looking at that exhibited high flexibility: the tryptophan, threonine residue, as well as a leucine 167. Simple distance restraints. This is the convergence, just to give you an idea that they were very well converged simulations. And then if you look at the free energy plot, you see that in the wild type, you have A1 and A2 regions, as well as A3 region, but which is less. And we could see the three conformations of the tryptophan, the intermediate conformation, the flipped out conformation, and the flipped in conformations. Although the flipped out conformation was not so much observed, so as the crystal structure pointed out. So we got this. But then, when the ligand was bound to it, and when we calculated the free energy surface when the ligand was present, we saw completely different structures. And in this structure, what was happening is that in region 1, if you remember, that helix to helix was completely being split open, and it was becoming much more mobile. So what we were seeing is that the structure was completely, the alpha network, the network of hydrophobic residues that is present in those regions were completely being abolished, so which resulted in higher flexibility. When you look at the single mutants that we created, we were observing a similar effect that we saw with the ligand. So clearly, a single mutant that was present was adopting a structure that was being adopted was exactly similar as when we saw that the drug was how the drug was behaving. So basically, if you get rid of that hydrophobic network, you see a similar behavior. When we also designed a double mutant, we got a similar response. So nothing very different. So how do you go ahead and test them? So the only way you test them is by doing some experiments. So we went on and we did these antibiograms. And we saw that if you have a mutant, you would see that the antibiotics are ineffective. But if you have in the wild type, the antibiotics play some role. But when you design these mutants, the single mutants as well as the double mutants, you see exactly the same effect as you would see that you could recover the antibiotic sensitivity to again. Again, we carried out some inhibitory concentration, which gives us exactly the same results that how we can retrieve the sensitivity, beta-lactam sensitivity. So finally, in conclusions, we identified some hydrophobic networks that are essentially needed for the function of the enzymes. The enzyme does not function if you have any of these broken down. So the mechanistic function is communicated by these hydrophobic networks. So what we think is happening is that there's some sort of communication that is taking place between the upper half or the lower half, or within the two domains that are essential for the function of the enzyme. So the loss of function mutants that we designed, as well as the drugs, they disrupt the alpha network in the same manner, exactly in the same manner. And of course, there's an allosteric pocket that is formed because of the flexibility as we see in these. So the strategy, so moving forward, our strategy is basically to identify and target pockets that disrupt hydrophobic network. This can be completely distinct from the catalytic site. So even if the catalytic site is active, you can somehow inhibit the mechanistic function of the enzyme by targeting these networks. And finally, to all our collaborators, the graduate students who did the work, Lee Woodford, who gave us the bugs to do the experiments, as well as Jess Healy and Francesco Gervasio. Thank you very much, and I'll take questions.